When you look across the Nigerian rainforest-savanna gradient, you see a vast, lush expanse of green. Traditional conservation operates on a simple baseline assumption here. If we can count a large number of healthy-looking trees, the ecosystem is safe. This reliance on visual surveys dictates how global environmental auditing works. Policy is written and ecosystems are graded based almost entirely on what we can physically see. Take the pterocarpus tree, known as African rosewood, a premier timber species with immense economic value, currently facing a massive scale of industrial logging and extraction. Or consider Piper guyanense, the West African black pepper. It is a vital ethnomedicinal plant and a spice crop that is essential to local food security across the region. When surveyors look at Piper plants growing in deforested farmlands, the crop appears incredibly robust. To survive the intense heat of an exposed agricultural field, the plant clears much thicker, rougher leaves. Meanwhile, if you measure pterocarpus trees growing in the wet rainforest and compare them to those in the dry savanna, they display 100% identical physical traits. To an auditor, they look completely uniform. This outward physical fitness creates a disconnect, a phenomenon known as the abundance paradox. It is a state where a plant's visible adaptation to its environment can actively mask a hidden divergence in its underlying biology. To measure the true health of these populations, we have to move past the phenotype, the physical appearance of the plant, and analyze the genotype, its DNA. If we plunge past the thick, tough exterior of a farmland piper leaf and examine its cellular structure, the data tells a different story. The tough texture found in farmland piper is a localized defense mechanism, an anatomical shield against the high light intensity and water loss of an open farming system. This chart compares the genetic diversity of forest piper against the farmland population, revealing a 12% drop. Biologists call this a genetic bottleneck. These agricultural zones act as genetic sinks, draining the variety needed to survive. This adaptation creates a false sense of security. The plant may survive today's heat, but its eroded genetic base leaves it vulnerable to a single new disease or a shift in the local climate. We see a similar deception in the pterocarpus timber trees, which appear identical no matter where they grow in Nigeria. This mechanism lies in their nuclear DNA. Wind blows pollen across vast distances, mixing genetic traits between distant tree clusters to keep their appearance uniform. But their maternal DNA, the genetic code stored in the plant's chloroplasts, is transferred solely through heavy seeds. These seeds drop directly to the ground and do not travel. This topographical map of Nigeria shows the consequence of restricted seed movement. The maternal DNA has fractured into 3 strictly isolated, distinct genetic zones, hiding inside these identical-looking trees. Biologists classify these separated populations as evolutionarily significant units. This exposes a flaw in how we manage logging. If we clear-cut one region, assuming the trees are identical to those in the next, we permanently wipe out a unique maternal history that took millions of years to form. Relying on site alone can lead to an extinction debt. We end up preserving a population that appears healthy today, but lacks the hidden diversity needed to adapt to future threats. So, in a heavily farmed and fragmented landscape, where is the high-quality DNA actually surviving? The most diverse DNA is surviving in institutional botanical gardens and locally managed traditional sacred groves. These culturally protected areas function as genetic refugia. They shelter the rare alleles and evolutionary potential that have been lost in the surrounding farmlands. To protect these remaining strongholds, global conservation frameworks and policies, like Nigeria's National Biodiversity Strategy, must change how they track data. We have to move from morphological auditing, the practice of simply counting tree shapes, to molecular monitoring, which tracks the deep genetic data. To save tropical ecosystems, conservation must evolve past just saving the physical forest. We must protect the invisible genetic code required to rebuild it.